Hi, Michelle Glass here, and welcome to our final lecture in our Chapter 3, Lecture 3 series. And our topic for this presentation is mitosis. Mitosis is the process of how a cell divides its nucleus. Now, there are four main stages. You have prophase, metaphase, Anaphase, and Telophase, and a really cool memorization device is PMAT to help you remember the correct order of these. So let us get started. So when we're talking about the cell doing division, what we're talking about really is, the cell's life cycle. And the cell has the same sort of life cycle that we have because, of course, it's just a miniature version of you. And so what we see is that most of the time the cell is in a phase that's described as interphase. So 90% of the time the cell is in this phase. Now, during interphase, you have different sections. So you have what's described as G0 in this phase. This is when the cell is just functioning, doing its job. So we're getting ready to talk about the integumentary system and the skin and the cells of your skin, and those cells are going to be doing the job of making your skin, right? And so they're going to be in the G0 phase. If you have a cell that's going to get ready to do mitosis or cell division, it's going to go into this preparation period. So we have what's called G1, we have S phase, and we have G2 phase. Now in these phases, this is when they're growing and increasing their number of organelles, preparing for division. The S phase is when they are synthesizing or replicating the DNA. So this is when sister chromatids are being formed. Now notice that the process of mitosis, which is the subject for this video, is only like 10% of its whole life cycle. So it's a really teeny tiny part. Sometimes when cells go into G0, they never go out. So some cells, once they're formed and they're mature, they do their job until they die. So sometimes cells don't go into mitosis unless there's injury or growth is necessary. So as we're talking about the division of the nucleus is, of course, where the DNA is housed. So we need to revisit this molecule. This will be the third time, at least, that we've talked about this molecule. DNA is, of course, your hereditary molecule. All of the DNA in your cell is referred to as your genome or genome. And the genome is organized during cell division into what are called chromosomes. So this is when that DNA molecule tightly wraps itself up around proteins. And the purpose here is so that you can easily transport the DNA without bits and pieces getting broken or lost. So just like when you move house, you pack up all of your belongings into a box, you wrap it up in tissue paper or bubble wrap so that nothing is damaged. Same idea here with the formation of those chromosomes. Now, the chromosome number is species specific. So in humans, we have 46 chromosomes, but other animals and plants have different number. So elephants have 56 and cabbage have just 18, but notice it's always an even number, and that's because you are getting half of your genetic information from your mother and half of your genetic information from your father. And so we see that your chromosomes are paired up, and we number them. So you have 2 number 1s and 2 number 2s and 2 number 3s and 2 number 4s all the way for humans until we get to that 46th pair. Well, excuse me, it's not 46 pairs. We have 46 total chromosomes, so that means we have 23 pairs. That was a big slip up, right? I just want to make sure you're paying attention. Okay, now when we talk about having our pairs of chromosomes, that complete set, half from mom, half from dad, we describe that as being a diploid cell. And we abbreviate diploid as 2N, 2 times the number that you got from mom, or 2 times the number that you got from dad. And the reason we use 2N instead of like 46 is, again, because the diploid number varies depending on the species. Now we're going to see diploid cells in our bodies. These are going to be called somatic cells. So for humans, in most other animals, these are all the cells of your body except for what we call the gametes or the sex cells. So the somatic cells are the cells of your body. They're going to be diploid, meaning that they have 23 pairs if you're a human cell. A haploid cell has half of that information. So haploid cells are represented by lowercase n for like whatever is that baseline number. You're going to see your haploid cells are going to be the gametes. Gamete is the fancy way of saying sex cell. So the egg cell and the sperm cell would each be haploid. Okay, so that when two haploid cells come together, you have a diploid cell, which is going to form that next organism. So when the haploid sperm, fertilizes the haploid egg, you get a diploid zygote, which becomes that next generation. Okay, so chromosomes would be the state of the DNA wrapped up in that package, preparing for division. Chromatin is the relaxed stage of the DNA. So anytime you're doing DNA replication or you're doing transcription translation, then you're going to be looking at that DNA in its chromatin phase. So during interphase, it's going to be chromatin. When you are replicating the DNA, this is happening, of course, in the S phase of interphase. And this is where you're going to go from one chromatid to two identical sister chromatids. So one is a photocopy of the other. And the signature of your sister chromatids is they will be attached to each other. So look here, I've drawn for you one chromosome, and I've shown where the centromere is located. And then we're going to replicate that during S phase. And so now we have two chromatids. They're attached at one centromere. They are identical to each other, but we still call this one chromosome because we count our chromosomes based on our number of centromeres. Okay? All right, we are now ready for PMAT, mitosis, prophase, metaphase, anaphase, and telophase. Okay, so starting out, we're going to show here what's called one homologous pair for our representation. So we have homologous pairs are not identical to each other because one of these is inherited from mom and the other one is inherited from dad. So hopefully they're coming from two separate gene pools, meaning that they're not closely related. So homologous pairs, that's like your two number ones and your two number twos and your two number threes. We said that in humans we have a total of 46 chromosomes, so that means we have 23 homologous pairs. Now when you have DNA replication, notice that you get the formation of your sister chromatids. So because we're going to take one cell and divide it into two cells, we need to take our one set of DNA, replicate it so we can then do a proper division. So notice that your sister chromatids are attached to each other and they are identical to each other, which I've tried to use the color coding to represent that. So here we still have one homologous pair, but we're seeing sister chromatids for the orange and sister chromatids for the green. Once you go through prophase, metaphase, anaphase, telophase, you get what are called your daughter cells. Daughter cells are your products. They should look identical to that original cell. So if that original cell had one homologous pair, then our daughter cells should each have one homologous pair. Now, mitosis is for the purpose of growth and repair. You're making a clone. So if you need more skin cells, you want to make sure you're replicating and making more skin cells and not something else. Okay, so let's actually go through each of these steps in detail. Notice that I'm drawing a cell that just has a diploid number of 4, so I'm just representing 4 total chromosomes or 2 pairs. I have a long pair and a short pair. I did this just so it'd be simpler to draw. So we are starting out, we're still in interphase. Now we're doing our DNA replication, so that's the S phase of interphase. So now that we see our sister chromatids, we're ready for cell division. Okay, so now we're ready for prophase, which is our first step. So in prophase, that nuclear envelope disappeared, the DNA condensed to form the chromosomes, and our spindle apparatus forms. The spindle apparatus has protein fibers that are going to help us move our chromosomes to opposite ends of the cell. Our second phase is called metaphase, and in metaphase, those spindle fibers are going to move those chromosomes to line up in the center of the cell. And then in anaphase, we have our spindle fibers contracting and pulling those sister chromatids apart, so we're going to opposite ends of the cell. And then in step 4, we have what's called telophase, and you can remember 2 new nuclei. This is essentially the reverse of prophase. So what we see is that that spindle apparatus disappears, nuclear envelope reforms, and those chromosomes are going to relax back into that DNA relaxed form, which is called chromatin. As telophase is finishing, we get another stage called cytokinesis. Cytokinesis is the actual splitting of one cell into 2 new cells. So when we do cytokinesis, we're dividing that cytoplasm and all of those organelles from one cell into 2 cells. And then that cell membrane is pinching off so that you have a distinct cell. So cytokinesis is starting here in telophase and then finishing up. Okay, so here we have our whole process. So we have our first cell here that's in interphase. We see our 2 homologous pairs, representing that they're not identical to each other, they're not attached. We have a long and a short pair. Going through S phase of interphase, we get DNA replication, so now we have sister chromatids. We can see that they are attached, they are identical, and we still have a long pair and a short pair. Okay, now we're ready for prophase, metaphase, anaphase, telophase. Here's a pretty cool memory device to help you with this. So you can put your hands together like this. Prophase is when the DNA is condensing to form chromosomes, so we're going to use our fingers to represent our chromosomes showing up. We can also remind ourselves that the nuclear envelope is going to disappear and the spindle apparatus is going to form. Next, we're ready for metaphase, which is when the chromosomes line up in the middle of the cell. Then we have anaphase, which is when those spindle fibers are going to contract and separate the sister chromatids, moving them to opposite ends. And then we have telophase, which is when we get the formation of 2 new nuclei. So nuclear envelopes reform, spindle apparatus disappears, and the DNA relaxes back into chromatin. All right, that's pretty cool, right? All right, let's finish this baby up. Okay, so mitosis, again, is replication for the purpose of growth and repair. You're making more of the same. Our daughter cells are identical to our original cell. So if our original cell is diploid, then our daughter cells are going to be diploid. If our original cell had 2 homologous pairs, our daughter cells are going to have 2 homologous pairs. Okay? All right, now cancer happens when the process of mitosis is happening outside of the rules that are set up by the cell. So it's basically abnormal or uncontrolled cell growth. You can have 2 different types of tumors. You have what's called a benign tumor or a malignant tumor. The benign tumor does not spread and can be fairly easy to remove, while the malignant tumor is cells are continuing to divide. So this is much more invasive. They can spread to other tissues, making it really difficult to pull those out. Cancers can start because of carcinogens. Carcinogens are mutagens, examples of carcinogens is what I should have said, include mutagens, which are chemicals that cause DNA mutations. Viruses can cause cancers, radiation can cause cancers, and other chemicals can cause cancer as well. And that finishes us up for our Lecture 3 series. Almost. I got ahead of myself. Here's what makes cancer bad. Cancer cells are uncontrolled cell growth. The cells are too large or they can be too small for their proper function. They can be really greedy about nutrients, so they can take nutrition away from healthy cells. And they can perform those normal functions for cells, but in an abnormal way, so it can cause problems that way. And now we're done.